Picture this. You're making ratatouille for the first time, but you do not like tomatoes. You decide to leave them out and finish the dish. But upon taking it out of the oven, you see that it has not congealed and is instead a soupy mess. What has caused this mishap? Similarly, there are many mysteries of the human body, and like the ratatouille, we do not always know what is causing certain diseases. However, transcriptomics are bringing us closer to the answer. But before we could begin to develop transcriptomics, we had to study the genome. The genome is a collection of the DNA present in the nucleus and mitochondria of a somatic cell. Since the start of the Human Genome Project, genome sequencing has developed rapidly. Genomic sequencing can give information on genetic variants that can lead to disease or can increase the risk of disease development. The transcriptome is the initial product of genome expression, as a sum of all of the organism's RNA transcripts. The two main contemporary techniques utilize microarrays, which are used to assay biological material and quantify a set of predetermined sequences, and RNA sequencing, which uses next-generation sequencing to show all sequences. But why are transcriptomics important? Analyzing RNA sequences and gene expression in their entirety allows researchers to determine when and where a genome is turned on or off in cells and tissues, and shows broad, coordinated trends that more specific assays are unable to accurately represent. While next-generation sequencing technology was essential in the development of transcriptomics, it does not provide phenotypic information, such as cell surface protein levels. And this is where cellular indexing of transcriptomes and epitopes by sequencing, or CITE-sequencing, comes in. This is a method in which oligonucleotide-labeled antibodies are utilized in order to integrate cellular protein and transcriptome measurements into a single-cell readout. DNA sequences are added as barcodes attached to cells in a sample. CITE-sequencing uses these antibodies to represent protein detection as a quantitative and sequenceable output. The oligonucleotide-labeled antibodies are used as synthetic transcripts that get captured in a majority of oligodeoxythymine-based single-cell RNA sequencing. Because of this, cells can be immunophenotyped with a possibly unlimited amount of markers and unbiased transcriptome analysis. This year, the record amount of oligo-linked antibodies is 130, as science company BioLegend has developed the total sequencing human universal cocktail composed of 130 oligo-linked antibodies, enabling massive screening possibilities. Tomatoes are an essential part of ratatouille, and the dish will not congeal without them. While we know what causes problems in a ratatouille, we must now discover how cells can cause disease and what marks a possible attack, with the help and development of transcriptomics.